Blue genes might be expressed in brains, green might be expressed in blood, and red might be expressed in heart. And there are two main types of epigenetic marks that we'll talk about, and these are the post-translational modifications of the histone tails and DNA methylation. And these marks are malleable and affected by the environment and are disrupted in many disease states. So as you know, the DNA does not exist alone in cells. Rather, it is wound around these proteins, called histones. Together, the DNA and histones make up the chromatin. And the chromatin is further packaged into chromosomes. And within that chromatin, there can be more open euchromatin or more closed heterochromatin. So the epigenetic marks play a key role in determining these chromatin states and in regulating gene expression. So they do not affect the DNA sequence, as I said before, and some marks shown in green here are activating marks. They're associated with euchromatin and gene expression. And other marks are silencing marks, shown here in red as histone marks or DNA methylation. And these are associated with more closed chromatin states and gene silencing. So now let's dive into DNA methylation. So some roles of DNA methylation or 5-methylcytosine are regulation of transcription, mediating genomic imprinting, X chromosome inactivation, and repressing transposable elements. So let's talk about how DNA methylation is placed, removed, and interpreted. So the writers of DNA methylation are the DNA methyltransferases, or the DNMTs, and they catalyze DNA methylation by transferring a methyl group to the 5 position of cytosine, and they use the SM-methylmethionine or SAM donor, and this occurs at CpG dinucleotides classically, although more recently we know that there is non-CG methylation, but I won't really be talking about that today. And the DNMTs are a family of enzymes, mainly we think of these 3, and there certainly are others, some of which are actually catalytically inactive. But DNMT1 is the maintenance methyltransferase, and we'll talk a little bit more about that on the next slide. And DNMT 3A and 3B are the de novo DNA methyltransferases. And each of these has multiple isoforms, and they're highly conserved in the catalytic domain region, shown here in red. So this schematic will help us illustrate the process of placing, removing, and maintaining DNA methylation. So as we said on the previous slide, DNA methylation is placed by the DNMTs, and then we'll see here that it's removed either passively or actively by TET enzymes. So here, just to orient you, here on this slide and throughout a lot of my talk, these black vertical lines indicate CpG sites. The white hexagons or unfilled are unmethylated, and the pink filled sites are methylated. And you can see here DNMT 3A and 3B, it's just shown as DNMT 3, but this accounts for both 3A and 3B, catalyzes de novo methylation of unmethylated CpG sites. And it can act on hemimethylated sites, but they're thought of as the de novo DNA methyltransferases. And DNMT3A functions more in somatic cells, and DNMT3B is important in the global remethylation during implantation, which we'll talk about later. So patterns of DNA methylation are replicated semi-conservatively. So similar to DNA replication, with each round of replication, what you see with methylation is that it gets diluted, and one strand becomes unmethylated, and because unmethylated cytosines are added, and this results in CpG dinucleotide or CpG regions that are hemimethylated, so methylated on one strand and not the other. And so then DNMT1, the maintenance methyltransferase, is recruited to these hemimethylated sites and restores DNA methylation in the case of where it was previously present. So this is maintained through cell division. Specifically, DNMT1 is recruited, so here I'm showing DNMT1 and then this whole larger pink circle includes the DNMT1 enzyme here. It's recruited to these hemimethylated sites by PCNA and UHRF1, and then UHRF1 allosterically activates DNMT1, allowing it to actually catalyze the addition of a methyl group. So how is DNA methylation removed? So one way it can be removed is passively. So if you can imagine, if you replicate the DNA and DNMT1 isn't recruited there, you can sort of just passively dilute out the methylation, and that we know happens in some cases. And for a long time, that was the only way we knew how this could happen. But then eventually, these TET enzymes were described, and it might seem long ago to you, but this is the kind of like one of the newer description of all these types of machinery that we'll talk about, and this happened back in around 2009 or 2010. And so these TET enzymes can actually catalyze the removal of DNA methylation. And then in addition, and then there's this intermediate that I'll talk a little bit about more on the next slide. So the TET enzymes are essentially the erasers of 5-methylcytosine. So they catalyze the erasure of this mark. They're a family of dioxygenases that oxidize 5-methylcytosine through a series of intermediates. So here you can see the DNA methyltransferase adds the methyl group to the 5-position cytosine, and then the TET enzymes remove it. So they catalyze they oxygenate, they're dioxygenases that oxidize 5-methylcytosine to 5-hydroxymethylcytosine, and then to 5-formylcytosine, and then to 5-carboxycytosine. And these two intermediates, 5-formylcytosine and 5-carboxycytosine, are then removed by thymidine DNA glycosylase, creating abasic sites that are then repaired by base excision repair. And this replaces the methylated cytosine with an unmethylated cytosine. So that is the process of removing DNA methylation. And the TET family of enzymes has 3 members, TET1, TET2, and TET3, again with multiple isoforms. But these are also highly conserved dioxygenases with overlapping functions. And the cysteine-rich region is also quite important for catalysis. And the CXXC domain, which is present in 2 of them, is important for targeting or recruiting these proteins. And those that don't have that domain are recruited and targeted by binding to other partners. And then the 5-hydroxymethylcytosine mark is an intermediate that we think is actually important in differentiation, in particular neuronal differentiation. And so that is a mark that is being actively studied by many right now. So we've talked now about how DNA methylation is placed and removed, but how is it interpreted? Or how does DNA methylation at gene promoters lead to gene silencing? And the answer is through readers of DNA methylation called methyl CpG binding domain proteins, which are shown here. And this family of proteins, they have methyl CpG binding domains, most of them shown in purple, that bind specifically to methylated DNA for the most part. MECP2 is probably the most well-known of these proteins. It's involved, it's the gene that when mutated causes Rett syndrome. This has actually recently been shown to have the ability to bind non-CG methylation, but for example, MBD2, another member of the family, it binds essentially only to CpG methylation. But regardless of that, they bind to the methylated DNA, and then they recruit these large repressive chromatin remodeling complexes through their transcriptional repression domains. And that combination of the DNA methylation and recruiting these of chromatin remodeling complexes leads to gene silencing. So how you kind of picture this happening is you start out with this actively transcribed promoter. And you can see that there's, we haven't talked about the histone marks yet, but there's histone acetylation, transcribed. The DNA methyltransferases come in, they methylate the DNA, they recruit these other chromatin remodeling complexes. So once the DNA is methylated, the methyl CpG binding domain proteins bind to the methylated DNA, recruit these complexes that also contain histone deacetylases, lysine methyltransferases, and other structural components, and that leads to more closing of the chromatin and gene silencing. So DNA methylation occurs at certain sites, and as we said, is associated with gene silencing. Again, here the same for these, the white or unfilled CpGs are unmethylated, and the pink ones are methylated. And so the CpG sites tend to be depleted in the genome, and about 70 to 80% of them are methylated. There are these regions called CpG islands that are approximately 1 kb in length, and they have a high density of CpG sites, so greater than 50% of CpGs are methylated. Or I'm sorry, are greater than 50% of the base of the CpGs is what I meant to say. They're found at many promoters of housekeeping genes, and they're generally protected from DNA methylation. And this protection is thought to occur through a combination of factors, but proteins that have CXXC domains can be targeted here, and then H3 lysine 4 methylation, a histone mark that we'll talk about later. These are some ways that it's thought that these CpG islands are protected from DNA methylation. But when they do get methylated, they lead to long-term gene silencing. And so this happens at imprinting control regions, genes on the inactive X chromosome, and hypermethylated tumor suppressor genes. And then beyond the CpG islands, there are these shores and shelves that sort of go out away from the CpG islands. And the shores are also thought to be involved in regulating gene expression. But this is just kind of showing a CpG island in an individual locus. But if we expand that view from one locus to larger genomic regions, this slide shows kind of more the genomic distribution of DNA methylation in the genome. So the top panel shows CpG methylation. You can see the y-axis here. So CpG methylation on the y-axis and is shown in aqua. And then the middle here is the CpG density, with light gray being a low density of CpGs and black being a high density of CpGs. And then this shows the percent of DNA methylation valleys that have CpG, where the CpG islands are. So if you look at the CpG islands, which are shown here in light green, these have the highest CpG density. You can see that, they correspond with the black, and the lowest DNA methylation. About half of these are located in the promoters of housekeeping genes, shown here in light blue. Others can be embedded within or at promoters of developmentally regulated genes, shown in the darker blue. And these developmentally regulated darker genes are located within these kind of DNA methylation valleys, which are shown in the dark green above them. And these have low levels of DNA methylation and variable CpG density. And so the idea is that these developmentally regulated genes, they are activated by transcription factors binding to enhancers, but that the chromatin around this is more facultative chromatin, so they're able to be expressed when needed and able to be silenced when not needed. And then you can see some repetitive elements here, which tend to be highly methylated and have fewer CpG sites. So any questions on kind of the basics that we've gone over so far? So then let's kind of move on to try to understand some actual biological processes that DNA methylation and other epigenetic marks are important for. So genomic imprinting is important, and it is really the definition is the differential expression of alleles depending on the parent of origin. So classical teaching is that most genes are expressed equally, more or less, from the paternal and the maternal copies. However, imprinted genes are different, and they're differentially expressed depending on the parent of origin. So back in the 1980s, McGrath and Salter and Serrani performed a series of nuclear transfer experiments that showed that both a paternal and a maternal genome are required for successful mammalian reproduction. So basically they took a mouse egg and sperm. They had, upon fertilization, if they removed the paternal pronucleus and replaced it with another maternal pronucleus, this was they called it gynogenetic lethal, so it was lethal. If they removed the maternal pronucleus and replaced it with another paternal pronucleus, so that there were two paternal pronuclei, this was also lethal. But if they removed the paternal pronucleus and replaced it with a different paternal pronucleus, or the maternal pronucleus and replaced it with a different maternal pronucleus, such that there was both a maternal and a paternal, then this was viable. And so this suggested that you needed complements from both the paternal and the maternal. And then many people went on to do many more studies, some using chromosomes or reciprocal translocations that suggested that it wasn't the entire genome of each that was needed, but that it was specific regions. And those turned out to be imprinted regions or imprinted genes. So I just kind of I have a couple of slides to kind of go through genomic imprinting in the organism. I think it can be challenging, so please stop me if I'm going too fast or if something doesn't make sense. But we'll just kind of start here. So we have, so throughout these slides, the female mouse is pink, male mouse is purple. So here, when you have fertilization, so you have the egg and sperm, they each have imprints on them. And so upon fertilization, those imprints are maintained during cell division on the corresponding paternal or maternal chromosomes. And these imprints are maintained in all cells of the body, including the embryo, the placenta, and the other membranes. However, in the embryonic gonad, these imprints get erased. And then during sex determination, or after sex determination in males, haploid sperm cells acquire paternal imprints, and female haploid oocytes acquire maternal imprints. And so then if we go on to the next slide, so then these imprints are then, once they're established in the germline, they go on, so what they look like is sort of in the maternal case, you have an imprint, for example, at a particular imprinting control region here shown in red, and in the sperm you have a different imprint on a different locus shown here. And then upon fertilization in the zygote, again, these imprints get maintained in all the somatic lineages. However, when they get erased in the germ cells so that they can then be passed on to the next generation. So imprints are acquired by one parental gamete and stably maintained in somatic lineages. And they're a consequence of inheritance, not sex, in somatic tissues. So during this process in mammalian cells, there are dynamic changes in DNA methylation. And in fact, there are these two waves of global demethylation and remethylation. in the life cycle. So one during gametogenesis and the other during early embryogenesis. So here, the DNA methylation level is indicated on the y-axis, and time in development is indicated on the x-axis. And blue indicates male and red indicates female. And so what you can see is that during gametogenesis, the primordial germ cells, you have erasure of the somatic DNA methylation patterns in these primordial germ cells and establishment of sex-specific germ cell DNA methylation patterns, including in imprinting control regions, right? Because these are the germ cells that will go on to make this zygote. And so the primordial germ cells do have this erased, and the new imprint is placed based on whether it is a male sperm cell or a female oocyte. Then at fertilization, you have the zygote here. You have another wave of demethylation. And in males, this, and here, I'll point this out first, is that at imprinted loci, so imprinting control regions, in this case, during embryogenesis, these are not erased. So there's this wave of demethylation that occurs everywhere but at the imprinted loci. Okay? And so in and then the wave of, and then after you kind of have this wave of demethylation everywhere except these imprinted loci, then you have this wave of de novo DNA methylation, which reestablishes global patterns. And I'm going to zoom in on this part on the next slide. And so just looking at the part here during embryogenesis, here we're going to look at 5-methylcytosine or 5-hydroxymethylcytosine. Remember, 5-hydroxymethylcytosine is what the TET enzymes convert 5-methylcytosine to 5-hydroxymethylcytosine. And so after fertilization, you see this wave of demethylation. In the male pronucleus, shown in blue, you see this very precipitous drop of 5-methylcytosine and a concomitant increase in 5-hydroxymethylcytosine. So this is now thought to be active DNA demethylation by the TET enzymes, because if you have a drop in 5-methylcytosine, you're going to get an increase in 5-hydroxymethylcytosine if that's being catalyzed by the TET enzymes. And however, in the female pronucleus, this occurs much more slowly and is thought to be passive dilution of DNA methylation. And then upon implantation, you have this, again, this wave of de novo methylation by DNMT3A and 3B. But remember, again, the imprinting control regions escape the demethylation, and then the imprints are maintained through this mechanism with DNMT1, UHRF, and this other protein as a bigger protein. So kind of putting it all together and summarizing kind of what we've talked about with genomic imprinting. First, you have the establishment of the imprints, which occurs in primordial germ cells, and that's shown here in green. So you have paternal imprints placed in males and maternal imprints occur in females. And I should point out that male imprints are placed prenatally, whereas females occur postnatally. So once these imprints are placed in the primordial germ cells, when you have fertilization, egg and sperm, then you have these imprints are maintained during active demethylation of the paternal genome, and during passive demethylation of the maternal genome. These are maintained in the somatic tissues. And then during implantation, here then you have erasure, but the imprints are maintained in the somatic tissues. So this is sort of the process and how this imprinting occurs. Yes.
So is this sort of like the only mechanism whereby epigenetic variability can be transmitted through generations? Like, is the only way to have, some specific methylation pattern come from like a grandfather to a grandchild or grandmother to grandchild, only methylation of imprinting control regions?
I mean, this is how it occurs, yes, but I guess I don't know if I would say the only, because there's, I think that is the way that what you're saying would happen. There can be diseases where you have actually, and we'll talk about these next time, where you have like a genetic change that causes epigenetic effects that can be passed on. But yes, it's like for thinking about imprinting and establishment, things like that, yeah. And this is in mouse. So I'll just say, all the things I'm talking about are in mouse, but it's very similar in humans. So a lot of course, the studies have been done mainly in mice. So, but yes. Any other questions? So now I want to move on from kind of the bigger process of genomic imprinting and talk about what are these imprints, what do the molecular changes look like, and how do they function to regulate gene expression, and then how does this go awry in disease. This is just kind of a generalized schematic, and molecularly, imprints are these epigenetic modifications that distinguish parental copies of a gene and are acquired by one parental gamete. This is, we think of DNA methylation, canonical imprinting. Certainly there are other marks involved because none of these marks occur in isolation. But there is some other imprinting that can occur in the placenta separately that I'm not going to talk about. And these occur at imprinting control regions, which are discrete genomic elements that are necessary to orchestrate the monoallelic expression of imprinted genes in a particular domain. Importantly, this is a cis-acting mechanism affecting a single genetic locus, though it can affect multiple genes. Imprinted genes are often clustered with a non-coding RNA, and they may modify long-range regulatory elements and act on multiple genes, but again, in one locus and in cis. And they play an important role in mammalian development. So often we kind of think of paternally expressed genes promoting, and just to kind of think about this moving forward, keeping this in your mind, paternally expressed genes tend to promote growth, and maternally expressed genes tend to inhibit growth. And the thought is that evolutionarily this would be the most beneficial thing, because if you're male, you want to kind of be the biggest, strongest, evolutionarily, the biggest, strongest organism. And maternally, evolutionarily, you want to have as many offspring as possible to continue the species. So you kind of would want to inhibit growth. So that's kind of the thinking of maybe one theory of why these kind of evolved this way. And so I'm going to just move on to show you, because this is just kind of a schematic to show that you have methylation involved in imprinting control regions. There's often the involvement of this non-coding RNA. There can be insulators that bind alleles specifically, and then you have differential expression of the genes. But now I want to now move on and show you an actual example of a locus. It's a bit simplified. We know a little bit more about the molecular events, but I think for teaching purposes, these are sort of the classic teaching about this particular imprinting control region. So Beckwith-Wiedemann syndrome results from altered genomic imprinting of genes on 11p15. In the normal situation, on the maternal allele, which is shown in pink, versus the paternal allele shown in blue, there are two imprinting control regions in this chromosomal region. So the first one over here is the KCNQ1 imprinted domain. So in this case, on the maternal allele at the imprinting control region, you have methylation, but at the imprinting control region on the paternal allele, there's no DNA methylation. So on the paternal allele, this allows for expression of this antisense transcript, KCNQ1 overlapping transcript 1, or LIT1 for short. But on the maternal allele, because of the DNA methylation, this antisense transcript is not expressed. And so the antisense transcript silences the genes in the region on the paternal allele, but on the maternal allele, these genes can be expressed. So what does that do? So focusing on CDKN1C, this is a cyclin-dependent kinase inhibitor, which is a growth inhibitory gene. So what that means then is on the maternal allele, this growth inhibitory gene is expressed, but on the paternal allele, it is not expressed. Okay? Then there's this other imprinted domain, the IGF2H19 imprinted domain. And in this case, on the paternal allele, at the imprinting control region, you have DNA methylation. On the maternal allele, there's no DNA methylation. So what this means is that this insulator CTCF can bind on the maternal allele, but not the paternal allele. When CTCF binds, the enhancers cannot loop back and activate IGF2, which is a growth promoting gene, insulin-like growth factor 2. So it's a growth factor, promotes growth. So on the maternal allele, this is not expressed. On the paternal allele, however, you have the DNA methylation, which prevents CTCF binding and looping, and then it allows for the enhancers to loop back and activate IGF2. So on the paternal allele, it is active. So in the two different loci, you have on the maternal allele an active growth inhibitory gene on the maternal allele, and you have an inactive growth promoting gene. Whereas on the paternal allele, you have an inactive growth inhibitory gene, but an active growth promoting gene. Okay, so that is sort of a way to regulate growth. So what happens in Beckwith-Wiedemann syndrome? So Beckwith-Wiedemann syndrome is an overgrowth condition. So there are many ways you can get Beckwith-Wiedemann syndrome. We'll only talk about 4 today. So for Beckwith-Wiedemann syndrome, you can have loss of methylation at this imprinting control region, the KCNQ1 imprinted domain. So if you have loss of methylation here on the maternal allele, then you express the antisense transcripts from both, and this silences CDKN1C on both copies. So you're now not expressing the growth inhibitory gene from either copy. Okay, so that doesn't inhibit growth, so you get overgrowth. Another way you can get Beckwith-Wiedemann syndrome is you can have gain of methylation at imprinting control region 1, or this IGF2H19 imprinted domain. So if you have gain of methylation on the maternal allele, then you have biallelic methylation. CTCF doesn't bind on either copy, and you can have looping and expression of IGF2 from both copies. So here you have a growth promoting gene that, rather than being expressed on one allele and not expressed on the other, it's expressed on both. So that's another way you can get Beckwith-Wiedemann syndrome. Yeah?
Does methylation always result in expression of that maternal-paternal set of genes? So in this case, it's methylated at those 2 sites, and so we have expression of that loci.
Right, so it's sort of like not the methylation itself that's causing expression here, it's the methylation of the imprinting control region that's preventing the transcription of this, but allowing for the expression of the other genes. Mm-hmm. So you're asking, is it always there? Like,
is it sometimes that the methylation results in the loci not being expressed, the entire region?
No.
Okay.
No, not. I mean, it's not really what we think happens, but you can have, and I won't talk too much about this, but you can have like deletions or duplications, any kind of sequence change in there that adds space or removes space you could imagine could disrupt this whole process. So I guess you could maybe imagine a time where if you had like a deletion here and the methylation got moved more closely to this promoter, that maybe could it silence that gene, maybe, but that's not something that we know of at this point. But there certainly are deletions and duplications that can occur that kind of mess up the spacing, which is really important here for all of this. Does that make sense?
Yeah.
So now, another way you can get Beckwith-Wiedemann syndrome is you can have essentially both of these at the same time. Both of these. I'm going to show you what I mean. So you can have paternal uniparental disomy of this region of the chromosome. I don't know if you've had this, but basically paternal uniparental disomy, or any uniparental disomy, means you basically have two copies of one parental chromosome, or maybe not the whole chromosome, but this region. So in this case, you have two copies of the paternal chromosome, and that can essentially lead to Beckwith-Wiedemann syndrome by both of the mechanisms we talked about before, because you only have the paternal. So you're not going to express CDKN1C from either copy, and you're going to express IGF2 from both copies. Does that make sense? And that can happen from like a trisomy rescue. So if you have a trisomy and then, the organism sort of gets rid of one of the chromosomes, then you can have this. That's how paternal uniparental disomy occurs. And then the last way that we'll talk about that you can get Beckwith-Wiedemann syndrome is you can actually have a genetic mutation in CDKN1C, but that only matters if it's on the maternal chromosome, because if it's on the paternal chromosome, that gene isn't expressed that gene isn't expressed anyway, and so it doesn't actually matter if you have a mutation of CDKN1C on the paternal chromosome, but it matters if it's on the maternal. And this is a very low percentage of Beckwith-Wiedemann syndrome is caused by this mutations in this gene, but if there's a family history of Beckwith-Wiedemann syndrome, it's about 40% of cases. So just to kind of summarize the different molecular etiologies that we talked about, the most common is loss of methylation of imprinting control region 2 on the maternal chromosome at the KCNQ1 locus. That's about 50%. About 20% result from the paternal uniparental disomy at this region. Only about 5% gain of methylation on the maternal chromosome at the IGF2H19 locus, and then about 5% of cases result from mutations in CDKN1C if they're not inherited, but about 40% if they are inherited. And then some of these other things that are listed aren't really relevant for you all, but you can, like we were just discussing, like if you have a microdeletion or a translocation or an inversion or a duplication, something that messes up the spacing in the region, that can also cause Beckwith-Wiedemann syndrome, but each of those by itself is very rare. We now know of diseases where you can have methylation abnormalities at multiple imprinting loci at once, and there's a very severe form of Beckwith-Wiedemann syndrome where you can have mosaic genome-wide paternal UPD. So you basically have paternal UPD of all the chromosomes, but it's mosaic, because if it wasn't, it wouldn't be compatible with life. But those individuals have very severe phenotypes. So what is the phenotype of Beckwith-Wiedemann syndrome? So these individuals, as I said, it's an overgrowth condition, so they often have macrosomia, or large increased weight at birth. We used to call it hemihyperplasia. The new term is lateralized overgrowth, so meaning one side of the body is bigger than the other. They can have macroglossia, which you can see here in this picture. They can have anterior abdominal wall defects, so meaning on the abdomen. They can have a range of defects. The most severe is an omphalocele. In that case, they're born with the abdomen open and the organs are outside of the body, and they have to be surgically placed back in. They can have an umbilical hernia, which is just basically a belly button that sticks out. And then they can have diastasis recti, which is kind of it's the abdominal musculature not closing completely, but everything's still inside. And then they can also have hypoglycemia, or low blood sugar. This can be due to hyperinsulinism. It can be either prolonged or transient in the neonatal period. They can have organomegaly of the kidneys, liver, even the spleen. They can have ear anomalies, which include anterior linear earlobe creases like you see here. They can also have posterior helical pits, which are just like little divots in the skin on the posterior part of the ear. They can have this facial nevus simplex, which you can see here, this red birthmark. And then they can have polyhydramnios, which is too much fluid, or a large placenta. Just think about everything being too much, like overgrowth, so making too much fluid, having a large placenta. They can also have congenital anomalies like cleft palate and structural heart defects. But the most important thing for individuals with Becker-Wiedemann syndrome is that they are at increased risk of tumor development, about 8% overall. And this includes embryonal tumors like Wilms tumor of the kidney, hepatoblastoma of the liver, and some others. And so for this reason, in the clinic, when we have an individual with Becker-Wiedemann syndrome, we do tumor surveillance, and they get abdominal ultrasounds every 3 months until age 8 years. And they get alpha-fetoprotein, and that screens for the Wilms tumor. It's basically looking at the kidneys to see if there's any tumor, because of course, if you detect something like that early, it's a much better prognosis. And then they get AFP or alpha-fetoprotein levels, which is something that can be released from the liver if there's a tumor growing in the liver. So if they have hepatoblastoma, we would see that on this blood test. So they get these both every 3 months, either until age 4 or age 8. And the reason we stop at those ages is because at those ages, their risk for developing those particular tumors matches that of the general population, and we don't screen every child for these tumors. So once they're basically, as they're younger, they have more of a risk of developing tumors, but then as they get older, that risk goes away. So, and they generally don't have cognitive issues, although there are some subtle neurobehavioral findings that we've seen in our clinic. So that's kind of the big imprinting disorder that I wanted to spend time on, so that you can actually see how these things can go awry and sort of what the molecular events look like. And I mean, we send tests in the clinic where we look at DNA methylation. We send a high-resolution copy number array to see if there are any deletions or duplications, and we also do CDK and Lindsay sequencing. So this is totally relevant to what we do in our clinic to test these patients. So I wanted to talk briefly, because I think this is just a really interesting kind of set of disorders. So Russell-Silver syndrome, or Silver-Russell syndrome, depending if you live in the US or Europe, results from altered genomic imprinting also. And it affects this locus. A subset of Russell-Silver, most Russell-Silver, results from disruption of imprinting at this locus as well. And it's sort of the opposite of Becker-Wiedemann syndrome. So Russell-Silver syndrome has the opposite phenotype. It is a disorder where they are very small, short stature and failure to thrive or reduced weight, but they have a normal head size. And so in these individuals, it's essentially a growth deficiency. And so in these individuals, they have paternal hypomethylation of the H19 IGF2 locus. And so that means that without that methylation, the insulator CTCF can bind, and it prevents the looping of the DNA to activate IGF2. So on both the maternal and paternal chromosomes, they're not expressing IGF2. So that's the most common form of Russell-Silver syndrome. And just to illustrate the point, is the opposite of Becker-Wiedemann syndrome. So that type of Becker-Wiedemann syndrome, you have both copies expressing IGF2, and Russell-Silver, you have neither copy expressing IGF2. And again, features of this disorder are pre- and postnatal growth. We don't really, are going away from the term growth retardation, growth deficiency. They still have a typical head size, so that means they have relative macrocephaly, because if they're very, very short, but their head size is appropriate for age, it's relatively large. They have severe feeding difficulties, body asymmetry as well. They have recurrent hypoglycemia, low blood sugar as well. They have high-pitched voices, low muscle mass, excessive sweating. They can have male genital anomalies, precocious puberty, insulin resistance. They can occasionally have some neurocognitive problems, and they can have some structural anomalies of various organs. And they have this very characteristic facial appearance with prominent forehead, and this asymmetric face. You can kind of see this cheek is smaller than this. And then the face is like an inverted triangle, kind of with this large head and this very small chin. They have dental crowding, this kind of downturned mouth, and micrognathy is just a small chin. They can also have fifth finger clinodactyly, which is shortening of one of the bones in the finger, which makes it look like the finger is turned in. And then they can have some spots on the skin, cafe au lait macules, and then some shoulder dimples, which we see commonly. Then I just wanted to kind of touch on a few additional imprinting disorders. So in general, imprinting disorders, as we saw in back with Wiedemann and Russell Silver, that set of disorders really shows you how important imprinting is for regulating size, right? It's also important for regulating cognitive function. Prader-Willi and Angelman syndromes are kind of, again, disrupted imprinting of a different locus, the 15q11 locus. In the case of Prader-Willi syndrome, I want you to hear about these disorders because they're important in human genetics, but I also want to kind of just illustrate some patterns. So, a lot of these imprinted disorders, they can be caused by a deletion or UPD, either maternal or paternal, or an imprinting control region defect and methylation. So it's just kind of, they all have those possibilities, many of them have those possibilities. So those are, again, the same ways that you can get. I'm not going to go into such detail with all of these, but these are the ways that you can get Prader-Willi syndrome. And I like to remember it, Prader-Willi syndrome, no papa, meaning there's no paternal contribution. So you can have a paternal deletion. You can have maternal UPD, meaning there's two copies of the maternal chromosome, but no paternal. And then you can have an imprinting control region defect. And these individuals, in infancy, they have very low muscle tone, poor feeding, and failure to thrive, meaning they're small and have very low muscle tone and have developmental delay as well. Later in life, they continue to have developmental delay and intellectual disability, but this failure to thrive or low weight turns into, they develop this hyperphagia where they eat and eat and eat, and they don't feel full ever. So then they develop obesity and hyperphagia, and they remain short, and so they have this very short stature but obese. You can see this girl here. And they also have very small hands and feet and kind of characteristic facial features. So Prader-Willi syndrome, they have this bitemporal narrowing of the forehead. They have this small downturned mouth as well. And then the small hands and feet, and central obesity is common. Angelman syndrome is sort of the opposite disorder, but they have, so in this case, they don't have the maternal contribution, so they can have a maternal deletion, paternal UPD, or an imprinting control region defect. They can also have a maternal UBE3A mutation, and they have intellectual disability, ataxia, which is sort of instability on the feet, a happy demeanor, and they generally do not speak, and they do have this fascination with water. So again, this is just showing you the genomic structure of 15q11 and the different causes, highlighting Prader-Willi syndrome here. So here you have the imprinting control region is shown by this blue and pink, and that regulates both the Prader-Willi genes and the Angelman syndrome genes. So the Prader-Willi syndrome part is shown in blue. It is normally unmethylated on the paternal allele, but methylated on the maternal allele. And that controls the expression of these genes here, including SNRPN and SNRNA here. And Angelman syndrome is sort of the opposite. But UB3A is imprinted with the brain, and so the imprinting control region controls this UB3A imprinting. And then these green genes surrounding all the other genes are biallelically expressed. So this is just an example. And then I did want to show kind of an example of a pedigree showing some imprinting inheritance. In this case, you have the dots indicate a UE3A mutation carrier, and then the blue individuals are affected with Angelman syndrome. So what you can see is that if you have a carrier, a paternal carrier, if that's passed on to anyone, any child, they are unaffected because on the paternal allele it's silenced. Okay, but if a maternal individual passes it on to her children, then they're going to be affected because it needs to be expressed in the maternal. And you can see that here as well. So, in summary, DNA methylation plays important biological roles. Writers, erasers, and readers regulate the levels of 5-methylcytosine. DNA methylation is restricted to certain regions. And genomic, we talked a lot about genomic imprinting. It refers to monolithic expression of a gene based on the parent origin. And the mechanism of imprinting disorders involves disrupted DNA methylation in cis at a single locus. Any questions about DNA methylation before we move on to histone post-translational modification?
During global demethylation, does it hit a 0% methylation, or is it just like most of it gets gone?
That's a good question. It's definitely drops off, and so I would say, yeah, I think it's generally thought to be like loss of methylation and then retaining of it. All right, so moving on to post-translational modifications of histone tails. And I will just preface this with, I've split this up into 2 different parts, but these marks all interact with one another. So we're just going to split them up to learn a bit about them, and then I'll talk a little bit about how they interact with one another. So just some basics. Chromatin is, as you know, composed of repeating units of nucleosomes that look like beads on a string, and this is kind of the famous electron micrograph showing that, published in Science in 1974. And the nucleosome has 146 base pairs of DNA wrapped around the histone octamer, about 1.7 turns. The H3, H4, so it consists of 8 proteins. And first, the H3, H4 tetramer is deposited on the DNA, and then 2 sets of H2A, H2B dimers are deposited. What do we know about histones? We know that they're highly conserved proteins, they're highly basic, and they're small, and they have this globular domain, globular domains with unstructured N-terminal tails. So what you can see again on this schematic of a nucleosome is that you have the globular domains within the DNA, which is shown in black, and then these unstructured N-terminal tails which kind of protrude out from the center. And these N-terminal tails of each histone get modified with epigenetic marks, which are shown here. And these modifications affect chromatin structure and transcription. So here you see the 4 histones that are the core histones. I'm not showing H1, which is the linker histone. And what you can see is that each histone, you can see its N-terminal tail here and its amino acid sequence. And what you can see is that each histone can be modified at various positions. And the modifications are shown on the side here. We can have methylation on different residues, and that can lead to either can be associated with active gene expression versus repression. Acetylation tends to be an activating mark, phosphorylation can occur, and ubiquitination can occur. And each of these can occur on various residues. And you can see that H3 is, from what we know, is one of the most modified histones. Certainly not all histone modifications are indicated here. So Yes.
Does the arginine methylation tend to be active or repressive normally, or is it?
It can be either. Yeah. I will say, I'll focus kind of more on the methylation. So methylation can be active if it's on particular lysines, and can be repressive, whether it's on a different lysine. So for example, methylation of lysine 9 is a silencing mark. Methylation of H3 lysine 27 is a silencing mark, but methylation of lysine 4 is an activating mark. So methylation is interesting because it can occur, and I'll talk about this on the next slide, in various forms. There can be mono, di, or trimethylation, and depending on where it is, it can be activating or repressive. But for example, acetylation is generally always thought to be an activating mark. So if we think about just the acetylation system, the writers of acetylation are histone acetyltransferases, or HATs. And then the erasers are the histone deacetylases, so they remove acetylation. And then the readers of histone acetylation are bromodomain containing proteins, which are shown here. And again, this is a key activating mark. It's either present or absent, so it's a binary mark, and it modifies all 4 core histones. For lysine methylation, the lysine methyltransferases place the mark, lysine demethylases remove the mark, and then chromodomain containing proteins tend to be the readers of this mark. And again, it can be either activating or silencing, and it's a quaternary mark, so it can be present or absent, and then if it's present, it can be mono, di, or tri form. And this tends to modify mostly H3 and H4. Yes?
So what do the readers kind of like do to the histones and in response to these different marks?
It's a great question. So you may remember for the DNA methylation, I've talked about the CPG binding domain protein. So that's a reader. So basically the readers we think of as binding, and you can kind of illustrate it here. So they basically bind to the marks, but then they recruit other complexes and then kind of determine what happens, because it's really these larger complexes that tend to determine, what type of chromatin is set up, if it's an active region or silent region. So yeah, so they're sort of interpreting the mark by bringing in these other components and doing that. So histone marks are associated with particular gene regions. So this is showing the histone H3 tail, and it's indicating some of the residues. So lysine 4, lysine 9, lysine 27, lysine 36, and lysine 79. And then underneath is a schematic of the gene, and it's supposed to represent an enhancer here, and then the gene here with the promoter here, and the 5'. And so what this is just showing is that certain marks tend to be present in certain regions. So for example, histone acetylation tends to be present at enhancers or promoters, that's shown in blue. Lysine 4 monomethylation tends to be more at enhancers, whereas lysine trimethylation tends to be more at promoters. And lysine 9 methylation can kind of be anywhere, and same with lysine 27 trimethylation. And so this is just kind of showing, and then H3K36 methylation tends to be within the gene body. And this just lists, this is definitely not a complete list, but it just lists some writers, erasers, and readers of each mark and kind of where they are in the gene, typically. And I don't necessarily expect you to know all of those things, but just thinking about they're not just anywhere. They're at certain places and they have their functions there. And so the other important concept is that these systems do not exist in isolation, as I said. The histone modifications and other epigenetic marks interact with and influence one another. And so this is just showing you some examples of this. And I'll start with this one over here. You can have histone H3 lysine 9 methylation. And if you have that, at a particular, on a histone, then that tends to inhibit the same lysine from being acetylated. Yet it promotes the binding of this reader, HP1. And then if HP1 binds, that then promotes H4 lysine 20 methylation, but prevents H3 serine 10 phosphorylation. And then the H4 lysine 20 mark can inhibit H4 lysine 16 acetylation, but can promote DNA methylation. And so you can see that if you have the H3 lysine 9, H4 K20, and DNA methylation, you get kind of this repressed state. And then over here, it's kind of the same thing. So it's just really to illustrate the idea that these marks do influence one another. We can learn about each one individually, but the reality is that they all talk, and this has been referred to as the histone code hypothesis. Another thing they can do is this ability to sort of influence one another and even influence within the same complex can lead to chromatin domains that can kind of self-propagate and recruit additional components. So this is, again, another key concept, which is that, so for example, H3 lysine 27 trimethylation. So this is placed by the PRC2 complex, which contains the catalytic subunit EZH2 and a reader subunit EED. And so if you have this complex, then EZH2 places the H3 lysine 27 trimethylation mark. It's then read by a member of the same complex, EED, so it's interpreted or read by EED, which then recruits more of the complex, which then, if you're recruiting the whole complex, you're recruiting the catalytic subunit, so then that's placing more of the mark. So you can see how you place the mark, you read the mark, you recruit more, and that's how these domains can propagate. And that tends to be a mark associated with facultative heterochromatin. We kind of talked a little bit about that earlier in the lecture about where developmentally regulated genes can be, where they can get turned on and turned off in facultative heterochromatin. Another kind of example of this idea of self-propagation and spreading is H3 lysine 9 trimethylation. So here you can have this SUB39H enzyme which places this mark, then HP1 binds to the mark, and then HP1 can then recruit additional things, additional components like DNMTs, and then that can lead to DNA methylation of histones, and so then it's this combination of sort of the H3 lysine 9 methylation and the DNA methylation, which kind of together really closes up this chromatin and kind of locks it into this silent state. And so this type of chromatin would be present like at pericentromeric regions and other regions of the genome that are silenced constitutively. And then I did put in a slide, and people may know this already, but I wanted to put in a slide about how do you study these marks. One of the main techniques that we use to study histone modifications is something called ChIP-seq. And I'm sure many of you have heard of this or even done it, but it's a commonly used technique to study histone post-translational modifications. And the idea is that you basically, you take an antibody to a particular histone mark, and first you cross-link the DNA and the histones, and you add this antibody, and the antibody then binds to the mark of interest, but because it's cross-linked to the DNA, it then pulls down the associated DNA, and then you can, the DNA is then released and sequenced, and then you can map it to the genome, and you can then, if you map it to a particular gene, for example, gene C, then if there are a bunch of reads that map there, then you can be, then that tells you that gene C has associated H3 27 trimethylation, if that's the antibody that you used. And if that mark is not present, when the antibody won't bind, and essentially no DNA will be detected, and you won't get any sequence reads, and so then you would say that this gene over here does not have H3 K27 trimethylation. So that's just a quick schematic of how ChIP-seq works. But you can also do ChIP-qPCR if you wanted to just look, say you wanted to know, like, is this mark present at gene C? You could do that and just do chip the chromatin immunoprecipitation part and then do qPCR to see if that gene is associated with the mark. So in summary from this part of my talk, post-translational modification of histone tails is a complex system with many layers. The histone marks interact with and influence one another. Chromatin domains and states can self-propagate. And then we talked a little bit about ChIP-seq being a technique used to study these modifications. Any questions? Yes.
We talked a bit about like mosaic embryos and like, you know, like having things like microlesions or kind of changes. I'm just, do you think that that could have an influence on like kind of epigenetic? Like I guess it's like if you have kind of like these slight changes in like dosage or like if perhaps like a region that should be like methylated isn't present in like a lineage for a while, like could that have an outcome on that phenotype later?
So can you say that again? So that if you have a mosaic embryo for...
Oh, sorry. Like we've talked a lot about like aneuploidy.
Oh, okay.
Yeah. Like I guess in that context, like if you have like small duplications or like small like deletions,
Mhm.
like do you think that would have could have like an interaction, I guess, with like changes in like epigenetic?
Yes.
Okay.
I do. So I will talk a bit about, I think that my lecture on Monday will talk a little bit about DNA methylation profiling in, so we're not going to talk about Mendelian disorders of the epigenetic machinery today, but that's the group of disorders that I work on and that I'll spend more time talking about on Monday. But basically the idea is that if you have a disruption in any component of the epigenetic machinery, that you then see a characteristic DNA methylation profile in blood in many of these conditions. We now know that it's not just epigenetic conditions that lead to these characteristic DNA methylation profiles. We now know that other genetic conditions, Down syndrome has a particular DNA methylation profile, we know now. In fact, there's even data that's either published or almost published where fetal valproate syndrome, which is this kind of like, I don't like to call it a syndrome, but it's this constellation of findings that can happen if a mother takes valproate during pregnancy. There's now a DNA methylation profile for that, which is fascinating because it's like, you can imagine that like a medication could have effects on epigenetics and how we can detect that. So yes, so my answer is yes. I think that because we know from, for example, Down syndrome that, you know, if you have trisomy 21, they do have a characteristic DNA methylation profile. So I do think that is, I think this is much bigger than what we initially thought it was going to be. Good question. Anything else? Oh, yeah.
So I mean, maybe this is a little bit of a tangent, but back to trisomy 21.
Yeah.
Is it a function of just the genes that happen to be on chromosome 21 that's affecting the methylation? Is it just the fact that there's more chromatin in general? What do you think leads to that unique methylation?
Well, considering that we see it even in monoallelic conditions, where, you know, not a trisomy, but if you have like a missense change in many of these components of the epigenetic machinery on one allele, we can see an entire genome-wide DNA methylation profile that's different. So you can imagine if you have a whole extra chromosome. So I think it's just these downstream, you know, any change really causes broad downstream epigenetic changes. But it's definitely, I would think it's just because all of these genes are, there's going to be differences in expression levels, and of course, you know, kind of it's just each change will have many downstream changes, and then that leads to this characteristic profile. All right, so we're done at noon.
Okay.
So we're going to go through this quickly. I just wanted to give another example of a disease state associated with different components of the epigenetic machinery. So cancer is a disease state associated with somatic mutations in components of the epigenetic machinery and disrupted epigenetic marks. On Monday, we'll talk about not somatic mutations, but germline mutations. But today, really many of these components of the epigenetic machinery were described first in cancer. And so I just want to illustrate some of these points of how epigenetic changes are involved in cancer. So I already said this. So this is just a schematic showing an individual and many different types of cancer. And then you can see all these various epigenetic machinery components, and they can have somatic mutations that lead to these different types of cancer. Okay, so how are these marks disrupted in cancer? Now, this is a very broad generalization, of course, but kind of the thought is that in a normal cell or in a normal epigenome, we think this is kind of what we think is going on, which I've told you some of today. So you have these CPG islands that are at the transcription start sites of genes, housekeeping genes and other genes that are expressed. So you have these white, unmethylated, highly CPG dense regions, CPG islands. They're protected from DNA methylation. The gene is expressed. This is a tumor suppressor gene. So you see all these activating marks, histone acetylation, lysine 4 methylation, these activating marks. You can have DNA methylation within the body of the gene. So that's typically what we see is like the DNA methylation that alters expression tends to be in the promoter. You can still have, and then in the normal situation, you have DNA methylation in the body of the gene, but not at the promoter. This is an expressed tumor suppressor gene. And then if you look outside of the genic region, so the non-genic flanking regions, in a normal epigenome, you see generalized repression. So you have H3 lysine 9 trimethylation, DNA methylation, closed chromatin state, and kind of this tighter closed chromatin. What happens over many, many years and many studies, what we've found is that in the cancer epigenome, however, you can get this, this hypermethylated promoter. So hypermethylation at the promoter, you see that now the CPG island becomes abnormally methylated. You have these closed chromatin marks, you have this closed chromatin state, you have nucleosomes over the transcription start site, and you have gene silencing. And so, of course, if this is a tumor suppressor gene, that's not good. And then what happens in the non-genic regions is that you have kind of hypomethylation of the DNA and more open chromatin. And it's thought that that can then lead to genomic instability and possibly oncogene activation. So this is kind of what we think of as kind of the broad changes in cancer. And many years ago, we were one of the first to look at histone modifications and how they relate to hypermethylated genes in cancer. And so this is just a very simple figure, and it's basically showing MLH1, which is a tumor suppressor gene. Here's the transcription start site. Here's the CPG island highlighted by these black bars. You can see the high density of CPG, and then these are PCR primers underneath. In one cell line, these are both colon cancer cell lines. SW480 has unmethylated DNA and methylated MLH1, and RKO has hypermethylated MLH1. And what you can see is that we know the gene is methylated. We identified the silencing marks being enriched at the hypermethylated promoter, shown in fill symbols, and then a depletion, and then a depletion of this mark using CHIP qPCR in the unmethylated copy, and then the converse for the activating marks. And then we did show that when you treat with this drug that inhibits DNA, that 5-azacitidine, which inhibits DNA methyltransferases, so you take that gene that once was hypermethylated and is now unmethylated, then all of these marks reverse. So that's kind of one example of what we can see in cancer. And then more recently, another mechanism of oncogenesis has been described that showing that somatic mutations in histones themselves contribute to oncogenesis. So this is a newer and more distinct, a distinct mechanism. So oncohistones are somatically mutated histones that lead to specific cancers. It's typically missense mutations that are located at or near sites of post-translational modifications. Many have been described on H3. And it tends to be a dominant negative mechanism. So classically, particular brain tumors have been associated with mutations at position 27 of histone H3, so it's usually a lysine, it gets changed to a methionine, and then others. And then certain types of musculoskeletal tumors, like chondroblastomas, have associated mutations at H3K36, where the lysine becomes a methionine. And again, I said this is a dominant negative mechanism. And so what the thought is that, I'm just giving one example here, is that if you have a mutation at H3K36 that changes it to a methionine, when the histone methyltransferase binds to try to methylate, it basically gets, it binds and it can't be released. And so what happens is it binds and can't be released, and then what you have is you can't place the mark that it's supposed to place, which is the H3K36 dimethyl mark. And that mark is supposed to be within gene bodies and intergenic regions. And so if you can't place that mark there, and you have all the component of the histone machinery that's supposed to place that mark there, tied up in these sites where it can't unbind, can't be released, what happens then is you have spreading of this other mark, the H3 lysine 27 trimethylation mark. And so what happens is you basically replace the areas where there should be H3K36 with H3K27, but H3K27 should be silencing genes at particular regions in H3. And so when you do that, you basically disrupt these broad domains of gene expression. And that leads to kind of this transition of one mark to another, and is disruptive and leads to these types of cancer. And it's now more common than was previously thought. Histone mutations are now thought to occur in about 4% of tumors. Many of them were missed. Histone, I think, mainly because histone genes are quite small, and they have many different isoforms. Mutations are found in all 4 core histones. It started out thinking that it was mainly in H3, but they're now found in all 4 histones, and they can be multiple histone mutations in many tumor types. So you can see how that can contribute to oncogenesis as well. This is just that proposed model of pathogenesis, where in genic regions you have polycomb group, which places H3K27 trimethylation, silencing this gene. And in intergenic regions, you have the histone methyltransferase, NSD1, 2, or SETD2, and this places the H3K36 mark. And this H3K36 mark kind of precludes polycomb spreading and silencing. But if you have dominant inhibition of the histone methyltransferase that places this H3K36 mark, and it's just bound and unable to place the mark elsewhere, bless you, then you can have, that can result in spreading of polycomb to the intergenic regions, and then de-repression of genes that are supposed to be repressed. So you can see how this would affect many different regions of the genome. And that's because these histone marks interact with one another. So it's a key concept that keeps coming up over and over again. I just listed here some FDA-approved epigenetic therapies for cancer because, as I said, many of these were described first in cancer. And so this is just to show that it has translated into therapies to use for cancer. And I'm just listing some of these and what phase they're in of clinical trials or approval. And then we'll end with some take-home points. So epigenetics plays important roles in many biological systems and processes. DNA methylation and histone modification systems are the main components. Both systems consist of these writers, erasers, and readers of epigenetic marks, which we'll talk more about on Monday. And the epigenetic marks interact with one another within and between systems to regulate gene expression and chromatin states. And many disease processes result from disrupted epigenetics. I've talked to you today about imprinting disorders, cancer, And then certainly we'll talk more about Mendelian disorders of the epigenetic machinery. And that these disease processes may be treatable by therapeutic manipulation of epigenetic marks. This is actually being used in certain types of cancers in the clinic, and we hope to also use that in the Mendelian disorders of the epigenetic machinery. And then those are just my references. Happy to take questions. Yes?
Are there any like treatments for the imprinting disorders?
That's a good question. No, not approved treatments. I would say Angelman syndrome is probably the closest one. There are clinical there are many drugs in development, and there may be some actual clinical trials that are recruiting, but not targeted treatments. So for all of the disorders that we see, certainly there are treatments, but it's like for Russell Silver syndrome, right? They're small, so you can give them growth hormone, and that can help with their size. It can help with their low muscle tone. It can help with their kind of body composition, with their low muscle mass, but it doesn't treat the underlying problem, and it doesn't treat all the other manifestations. Angelman syndrome, as I said, they are working on drugs that can sort of, reactivate the silenced copy and things like that. And actually I took care of a patient a couple of months ago here who was on a study drug for Angelman syndrome, but she was actually, I think, having an adverse effect of it. So, she was on a study drug from Boston Children's, and she was not doing well and was having worse problems than you would expect with Angelman, which is bad because it's a really bad disease. They are kind of thinking that it might have been a reaction. In fact, we did like an exome. Even though she already had Angelman syndrome, they were so concerned because she was having worse symptoms than we would expect that we actually did genome sequencing to see if she had a second condition. She didn't. But we can never rule out that she does have a second condition, and we just don't know it yet. But it seemed more like might be due to the drug. Other questions?
So is the mutation in the specific histone gene, what other sort of mediators of changes in sort of global methylation are proposed for cancer? Because those are only in like 6% of cancers, but almost all cancers have like global hypomethylation, specific hypermethylation.
Yeah. So your question is, what else does it?
Yeah. How else does cancer induce that epigenetic state?
Well, you can imagine there are somatic mutations, as I said, in various components of the epigenetic machinery. So that's one way. And then the histone mutations are another way. And then certainly there could just be like other genetic changes that have then downstream epigenetic changes. So cancer is, as you probably know, it's very hard because it's hard to know what the first thing was, right? Like you have many things are wrong. And you don't we think of these driver mutations versus passenger mutations and things like that, but it's harder to know what caused what because of when you detect it, it's usually there's tons of, like you said, tons of epigenetic changes, there's tons of genetic changes, there's a lot of things that are impacting one another, which is why I think, I'm a little biased, our disorders, Mendelian disorders of the epigenetic machinery it's nice because we know what the first thing was. They have a mutation in this gene, and then whatever, it's like whatever happened is sort of a result. We don't always know like what's the direct effect and what's the indirect effect, but we at least know what the first change was. So it's kind of a cleaner, I would say, cleaner system to study. All right, great. Thank you all.
Sorry,
Tired.